Hi everybody. For our topic, we will discuss about instrument named Fourier transform ion cyclotron resonant mass spectrometry, also known as FTICR-MS. Part 1, Hardware.
Let's begin with the hardware of Fourier transform ion cyclotron resonance mass spectrometry (FTICR-MS). It is a type of mass analyzer for determining the mass to charge ratio of ions based on their cyclotron frequency in a fixed magnetic field. Do you know how does the FTICR-MS work? The figure here shows the non-scale schematic view of a magnetic field-based trap with the electron spray ion source. The ion source produced at the atmospheric pressure are focused and accelerated to quadrupole, hexapole, and transported to the ICR cell. The vacuum system, not shown in the figure, consists of pumping stages down to the range of 10 to the power of negative 10 millibar in the ultra-high vacuum chamber of the ICR cell. The numbers shown are the proximal pressures in different pumping stages. The experiment will begin with the generation of ion in an electron spray ion source outside the detector. The ions generated are focused and accumulate in the hexapole. Radio frequency and a direct current are applied to the rods, accelerating the ions to the quadrupole. However, only the ions with exact mass to charge ratio can pass through at the particular radio frequency, while the other ions are being ejected. The selected ions are then passed into the ICR cell, which sits in the middle of a 7 Tesla superconducting magnet. The ions traveling in the magnetic fields are confined to circular orbits by the Lorentz force, which is shown by equation 1, and the process in this orbit at a frequency called the ion cyclotron frequency, which is given by the equation 2. To increase the detection efficiency and resolution, the ions are excited by a radio frequency pulse to a larger ICR radius, and the cyclotron frequency is then measured by detecting an image current that is induced as the ion pass by the detection plate. The resulting signal is called a quick induction decay (FID) that consists of a superposition of sine wave. Fourier transform of FID will convert the signal to the time domain, which is then converted to the mass spectrum after mass correction. This can be shown by the equation 3. Since the pressure in the cell is very low, the ion orbital motion can be maintained over many cycles, and the frequency can be measured with very high precision. So the FTICR-MS instruments can therefore be used to generate exceptionally high resolution spectra with great mass accuracy.
Part 2, the advantages and disadvantages.
So now, let's look into the advantage and disadvantage of FTICR-MS. FTICR-MS has high resolving power, which is greater than 500,000. It is by far the highest resolution MS technique available. It also has high mass measurement accuracy, which is better than 1 ppm. These two factors allow it to achieve identification with high precision. The mass resolution in MS is used to indicate its ability to distinguish peaks. The higher the mass resolution will provide more separation. In this FTICR-MS, the resolving power increased linearly with increasing applied magnetic field induction, which is B. Since a superconducting magnet is used in FTICR-MS technique, it is very useful in the analysis of complex mixture. For example, The applications in petroleomics, dissolved organic metal, metabolomics, and also proteomics. By using this MS technique, the narrow peak width allowed the signals of two ions of similar mass to charge ratio to be detected as distinct ions. The sensitivity is also enhanced, which we can distinguish between analyte and background species at the detection limit or even near the detection limit. These advantages can help to reveal new spatial detail from multi-image analysis. Besides, FTICR-MS can detect different ions simultaneously instead of one at a time. This is due to the detection signal in the analyzer cell typically lasts for several seconds, which results in numerous cycles of cyclotron motion. Thus, high speed of analysis can be carried out. On the other hand, the disadvantage of FTICR-MS is that it performs at slow data acquisition rate in order to achieve high resolution and mass accuracy. The ICR resolving power is the number of ion cyclotron orbits during the data acquisition period. There's one approach to decrease the required signal acquisition time, which is to use stronger magnetic fields. The increased magnetic field results in increased detector frequencies and therefore increase the achieved resolving power for signal acquisition of the same duration. Besides, FTICR requires superconducting magnet because its performance improves in proportion to the strength of the magnet, which is mentioned before. The magnet also serves to confine ions in a plane perpendicular to B at the most homogeneous spot located at the center of the magnet. Magnet need to be large because ICR dynamic range and upper mass limit also scale as the diameter of magnet ball. Therefore, it brings up one limitation, which is FTICR-MS is expensive and require high operating and maintenance costs. Usually, a large superconducting magnet accounts for as much as half of the total instrument cost because the liquid helium and nitrogen cryogen required.
Part 3. The application of FTICR-MS.
Okay, for the application of FTICR-MS has been used as one of the most powerful tools to decipher, or in other words, break the natural organic matter molecules. In FTICR-MS analysis, the matrix effect caused by the co-occurring inorganic substance in water sample greatly affect the ionization of NOM molecule. The inherent complexity of NOM may hinder its component classification and formula assignment. Next, FTICR can use for the macromolecules. In FTICR-MS, resolution can be improved by increasing the strength of the magnet. The useful signal is extracted from this data by performing a FT to give a mass spectrum. FTICR-MS can achieve higher level of resolution than other forms of mass spectroscopy by using a superconducting magnet. From this FTICR-MS, the complex mixture can be analyzed as the resolution allows the signal of ions with closely similar mass to charge ratio to be detected as distinct ions with their attomol sensitivity. Next, FTICR-MS can use for the identification of archaeological lipid residue. It is a high-resolution and sensitive method with low limits of detection capable of identifying lipid compounds in a small concentration, thus providing a highly potential new technique for the analysis of degraded lipid component. Next, FTICR-MS can use for metabolomics. Metabolomics is defined as the comprehensive quantitative and qualitative analysis of all small molecules in a system. Direct infusion of FTICR-MS is used in non-targeted metabolomics to obtain higher resolution snapshots of the metabolic state of a system. Next, FTICR-MS can use for biomarkers. Especially important are minimally invasive tests that require only blood and urine sample. This technique that have developed appear to be effective in the analysis of multi-FTICR-MS data. Next, FTICR-MS can use for proteomics. Proteomics is the study of protein expression in an organism. The use of high resolution of FTICR-MS, including upfront multidimensional liquid separation for the top-down intact protein levels or the bottom-up, which is the peptide level proteomic approach. The application of FTICR-MS is used by the researcher to analyze a sample of a complex mixture such as petroleum in solution. The ultra-high resolving power and mass accuracy of FTICR-MS allow the scientists to determine the elemental composition within even the most complex sample with a high degree of confidence. Traditional analysis performed with a variety of Fourier transform mass spectrometers offer decreasing resolving power and confidence in assignments of the elemental composition at higher mass-to-charge ratio when studying a broad MZ range.
We also provided the extra information about FTICR-MS in the next few slides.